Are we good?
Yeah, we're good.
Okay, fantastic. So I'm really happy to talk to you guys today about a question. And are my slides advancing?
Yep.
Okay, fantastic. About a question that I've been thinking about since I was this girl over almost 40 years ago. And this is a picture that was taken of me. In the US, we have something called senior prom. And this was my prom date at the time. And this is a few months after I was diagnosed with MRKH. And in the States, there's a bunch of different treatment options for people with MRKH, and I was given two of them. One was to do dilation using a bicycle seat, and the other was to have a surgery called a McIndoe procedure, which they take a big piece of skin off your butt, and they create a vaginal canal from that. And at that time, it was a 2-week in-hospital process. And I could not fathom the idea of sitting on a bicycle seat to create a vaginal canal at the time. So I chose to have surgery. And in those 2 weeks in the hospital where I literally had to lay flat in bed, I couldn't sit above a 30-degree angle. As you can imagine, that's really fun for a 16-year-old. This one question kept permeating through my thoughts. I couldn't get rid of it. And that question was, how did this happen? And that sort of led to other questions like, well, how does a uterus develop? How did it develop improperly in me? And this question just kept going over and over and over again. And it's this question that I think led me to become a scientist and led me to try to think about how MRKH happens. And I'm going to share with you my thoughts on how MRKH happens today. And when you think about it, I'm so glad that Allie showed the film because Esther really puts this in perspective, all right? So one of the things that Esther says, and she talked about it a little bit in the film, but this is a quote that I have from her, is that we all have different noses and ears. Well, so why wouldn't we have differences in our reproductive tract? And I hope by the end of today, you're going to understand a little bit more about how genes are involved in development and how subtle changes and when these genes are turned on and turned off, where in the cell they're turned on and turned off, and for how long they're turned on and turned off can lead to these subtle differences between my nose and Esther's nose, between my ear and Allie's ear, and really understand that it's genes that are really the drivers of all of embryonic development. And so small changes in these genes can actually have big effects in developmental processes. And we're going to go through this a little bit here. So I like to think that Müllerian development, which the Müllerian tissues include the uterus, the cervix, and the upper portion of the vaginal canal, as well as the fallopian tubes. And I'm going to show you a little film about what happens when all goes well. And if you think about the genes as every single one of these players in the Boston and the Harvard orchestra, you can think about they need to be playing in concert. If somebody's a little bit off, you're going to be able to hear that. This is a film by Anneliese Littman.
We'll start at the 6th week of gestational development in the indifferent stage. The mesonephric or Wolffian ducts, which develop into the male anatomical structures, are present. Arising from the mesonephric ducts, the ureteric bud induces differentiation of the metanephros, which will become the kidneys. As we're focusing on development of the female reproductive structures, we will not show the alimentary tract, rectum, or the rest of the development of the kidney. The Müllerian or paramesonephric ducts first appear in the 6th week and arise from invagination of proliferating coulombic cells and elongate laterally to the Wolffian ducts. They then cross ventrally and direct medially, and in the 7th week descend to reach the urogenital sinus near the cloaca. The caudal tips of the ducts adhere to each other as they contact the urogenital sinus. In weeks 8 to 9, the ducts fuse and the septum degenerates, forming one tube, the primitive uterovaginal canal. In week 9, the Wolffian ducts regress, leaving remnants behind. In weeks 11 to 12, the contact between the fused Müllerian ducts and the urogenital sinus induces development of the sinovaginal bulbs, which in combination with outgrowth from the Müllerian ducts results in formation of the vaginal plate. proliferation continues at the cranial end of the plate, increasing the distance between the uterus and urogenital sinus. Canalization of the vaginal plate proceeds in a cephalad direction and is completed by the 20th week.
So you can see just from that short video that there's a lot going on during development. And each of these, that uterus is made up of millions of cells, right? So each of those cells has, thousands of genes within that cell, and they all need to play their own orchestra. And so when mistakes happen, and any mistake can happen, the tuba can, play mistakenly during the wrong time of a Brahms, of a Brahms song, and you'd get something that just really, you would notice that. And so I want to show you a little bit though about what this can look like. So this is a cartoon of an average uterus. So you can see here, this is if you were to continue this up, the ovaries would be out here, and the fallopian tubes. This is the cervix, and this is the upper portion of the vaginal canal. We are not showing you the lower portion of the vaginal canal here at all. It's missing. So this is what is an average uterus, but I purposely use the word average because because everybody has a slightly different shaped uterus, because all these genes need to turn on and turn off in a precise order, and a small subtle change can lead to subtle changes in these average uterus. We're on the other end of a spectrum with MRKH, where we are either lacking these uterine tissues or have severely underdeveloped uterine tissues. But if you notice, there's a big black space in between, and I'm going to show you a little bit about the spectrum of uterine development. So many women are born with what's called a T-shaped uterus. So the uterus is formed, but there's a really very little space for an embryo to develop. There's a septate uterus. Remember in the film, the two sort of uterine horns come together, and then there's this kind of tissue in the middle. Well, that's exactly what the septate is. So this tissue here never regressed. There's a bicornate uterus, where you basically have two uteri that's been formed, and some people with MRKH actually do have two fully formed uteri. Can also be a unicornate uterus, where you only have one of these uteri developing, and all the way to MRKH. And there's other sorts of differences along the spectrum. So you can see that subtle changes in how these genes are turned on and turned off in these millions of cells can give rise to a variety of phenotypes. And if you think back to Esther's comment, we all have different noses and ears, and we can all have different development of the uterus as well. So if you look at this and you think about this sort of graphic here, you're like, what is this? Well, this is a graphic of what's called a gene network. And gene networks work together much like sections in an orchestra. So you may have a flute section shown in this sort of cluster, a drum section over here. And these are genes that are implicated to be involved in MRKH by several large genetic studies. Genes that have circles that are really big, or genes that are really big, that means that they have a lot more networks. So they're interconnected with a lot of other genes that are contributing to MRKH. And we're going to talk about one of these particular genes called LHX1. So LHX1 stands for limb homeobox 1, and it's known to be involved in development of arms and the head and other processes. However, Richard Behringer's group is one of the really leading researchers on uterine development, and he uses the mouse to understand how these genes are turned on and turned off during development and what processes are important for that. So he asked what happens when you completely remove LHX1 from the Müllerian derived tissues shown here, and these would eventually become the mouse uterus. So this is looking at two wild type animals. So this is a typical mouse uterus, both as a neonate, so right after birth, and as an adult. You can see the ovaries here, and this is what a typical mouse uterus looks like. But when LHX1 is absent in these animals, you can see that the uterus is very underdeveloped. The ovary is still present, but there's very little uterine tissue. So this really recapitulates in a mouse model what MRKH can look like in humans. And this shows that there actually can be genes that when they're disrupted and there's no copies of these genes, and the Müllerian tissues can give rise to something that looks very much like MRKH. So this is a subset of genes that are involved in Müllerian development. If we again here look at our average uterus, there's a gene called disc large that when it's mutated causes no cervix or vagina. People who are born this way can also fall into the MRKH community. And these genes here are known to be directly involved in processes of Müllerian development, and mistakes in all of these genes that really cause defects in the female reproductive tract that look a lot like MRKH. We've also worked with Dr. Larry Lehman at Augusta University to understand the genetics of MRKH in people. And these are two studies by two early career doctors, Lacey Williams and Sasha McHale, who are really starting to narrow down the genes involved with MRKH using human studies. And in fact, this is looking at over 150 people with MRKH and their family members. And they're starting to narrow down a list of candidate genes that could be contributing to the MRKH phenotype in this cohort of people. So what I want to say is that MRKH can be genetic. It's complex phenotypically. Each of us has MRKH differently. And when you see something that's complex phenotypically, it indicates that the genetics is also going to be complex. There's not going to be one gene like LHX1 that causes MRKH. There's going to be multiple genes and multiple different pathways to how MRKH happens. Mouse and human studies implicate at least 18 genes. And they found these large structural variants, which are basically like big chunks of your genome is basically missing on one of your chromosomes that are more prevalent in people with MRKH. And these may cause basically to inherit a predisposing factor that also requires a second hit to really acquire MRKH. I'm going to talk through that with you guys. So this is a karyotype. Many of you actually had a karyotype done when you're diagnosed to make sure that you had 2 X chromosomes. So you have 2 copies of every chromosome in your body, and these are the chromosome 1 from your dad and chromosome 1 from your mom. So I think of the genome as the motorway. So if we take this portion of the UK motorway, we're going to look at the genome in that context. So let's say you inherit a large chunk of this chromosome is missing, okay? That's going to severely disrupt traffic. You're still going to be able to get through because you can still go through on the other lane, but man, it's a pain, right? There's a lot of cones around and driving slow, but you can still get through and you can still make the genes that are involved in this chromosome. However, if a random event happens during embryogenesis that, say, puts a pothole or a sinkhole in the middle of the highway, that means that any genes that were located in that region are going to be completely absent in the Müllerian-derived tissues, just like we saw when we knocked out LHX1 only in the Müllerian-derived tissues. And that shows you that you can inherit a risk factor, which is the deletion of this large region here of human chromosome 1, and also a random event that occurs during embryogenesis to lead to MRKH. And things like this can happen when you see aunts and nieces with MRKH and other sorts of events like that. So once again, if we go back to our big sort of orchestra of genes that are involved in MRKH, all of these genes here circled in blue are mouse genes that are involved in uterine development. And you can see here that all of these mouse genes, including LHX1, have strong connections with other genes that are implicated in MRKH through human studies. And so you can see how that subtle defects in one of these genes may not cause MRKH, but if you have multiple defects, say here in TBX2, and this gene here, and this gene here, these can accumulate, and you can have, the whole flute section is basically playing the wrong notes at the wrong time, and that can lead to MRKH. So as again, we talked that there are predisposing factors and other ways that you can acquire MRKH, and it can be a genetic condition. In addition, in the past 3 years, I've learned that there are 2 known cases where somebody with MRKH had a child through gestational carrier and through IVF and gestational carrier, and they had a child who also had MRKH. And so this is showing that there's actually direct transmission of MRKH in a dominant fashion, and that would be such that you would actually this would just be how it would happen. And this has been implicated and potentially known from studies by Daniel Garon's group in France for the past 10 years. And it's really why I started to look at genetics and MRKH, because I realized that these studies weren't being talked about in the gynecological sphere, because gynecologists tend not to really look at genetics, which is kind of surprising, but gynecologists really tend to focus on the Müllerian development component and not necessarily the genes that are involved in Müllerian development. So I think it's time to include genetics in the MRKH clinic, and we're working here in the States to try to make that happen. 5% of people who've been studied by MRKH genetic studies have one of these large chromosomal deletions, and indicating that they actually have a risk factor for passing on MRKH to their children. So the American Society for Reproductive Medicine has this awesome tool. You can click on this QR code here, and you can see all the different variations in Müllerian development. It's really cool. I encourage you guys to look at it. It's highly interactive. And I really want to end my talk by thanking my very dear friend Meredith Brooks, who had MRKH. She was the founder of the Mid-Atlantic MRKH Foundation, and she also had a lot of other. Sorry, that was my timer. She had a lot of other underlying health conditions too. She had a very severe form of MRKH, and she found out that she had MRKH at 3 and had been in the hospital and had a ton of different surgeries. And sadly, she passed away. It'll be 5 years this September. And so the Beautiful You Foundation set up Memorial Travel Awards in her name, where we provide scholarships for people to attend MRKH meetings. And I just want to thank our team. These are all the volunteers who work for the Beautiful You MRKH Foundation, and I'm happy to take any questions. And I'm going to stop sharing so you can actually see me. So.